Bismillahirrahmanirrahim. Today our topic is cell fractionation. History or discovery. Cell fractionation process was discovered by Behrens in 1937. What does the cell fractionation mean? Cell means cell parts, which include cell wall, cell membrane, cytoplasm, and nucleus, while fraction mean to separate and shan mean process. So the definition is the process by which we separate parts from a cell is called cell fractionation process. Then here comes the steps involved in the process of cell fractionation. In cell fractionation, 3 steps are involved. First one is grinding the tissue. Grinding the tissue means separation of cells from tissues by the action of pectinase enzyme. Here we take a tissue and apply a pectinase enzyme, and then this tissue is divided into cells. Then second step is homogeneate formation. A cell is taken in a beaker with a proper osmotic gradient. The proper osmotic gradient includes proper pH, water, proper temperature, and pectinase enzyme, which is used for cutting. In homogeneate formation, a cell is taken in beaker with proper osmotic gradient, and pectinase enzyme cuts the cell wall and cell membrane, thus converting the cell into its part, and these parts are called as homogeneate. What is homogeneate? The cell which converted into its part is called as homogeneate. Here is a beaker. We take the cell which is converted by pectinase enzyme into the pectinase enzyme convert the tissue into cells. Then we take the cell into a beaker and provide the proper osmotic gradient, which is water, proper amount of water, proper pH, proper temperature, and pectinase enzyme is used to cut the cell wall and cell membrane and converting the cell into its parts. Then here is a third step which is called as centrifugation. In centrifugation process, transfer the homogeneate from beaker into the test tube. The homogeneate is transferred from beaker into the test tube. Put the test tube in centrifuge machine. In the process of centrifugation, we use a centrifuge machine, and then here is we take the test tube into centrifuge. Centrifuge machine starts spinning on following rates. That is 800 revolution per minute multiply gravity give us nucleus. 20,000 revolution divided by 15 minutes multiply gravity give us mitochondria. And here is 1 lakh revolution divided by 60 minutes multiply gravity gives cell wall, cell membrane, and microsome. Then here there is a new word called as microsome. What is microsome? So microsome consists of Golgi bodies, endoplasmic reticulum, lysosome, and peroxisome. Then here is 1,50,000 revolution divided by 3 hour multiply gravity give us ribosomes. Ribosome is the smallest organelle, so it give on the high revolution and divided by high time than other organelles. Then here is one sentence that centrifugation is the separation of the cell on the basis of density. So a nucleus is of high density, so it gives on the less revolution in less time. And ribosome, here is ribosome, which is the smallest particle, so which is the smallest organelle, so it gives the ribosome on the high revolution and divided by high time. And then in the test tube there are 2 types of solution. The homogeneate at the bottom of the test tube is called as pellet or gravity, while homogeneate at the top of the test tube is called as surfactant solution. The cut part of a cell. Then here came some MCQs like important points of this topic. The cut part of cell in a beaker is called homogeneate. Here is the main point is in a beaker. In a beaker there is homogeneate, while in a test tube there is we take the homogeneate to apply the test tube into the centrifugation process. The revolving machine used in this process is called a centrifuge machine. The enzyme used for cutting of the cell is called pectinase enzyme. Homogenate at the bottom of the test tube is called pellet or gravity, as we discussed earlier. That is pellet or gravity. The homogenate at the bottom of the test tube is called as pellet or gravity, while homogenate at the top of the test tube is called supernatant solution. So here these 2 points came. Then there is a point that centrifugation is a separation of cell on the basis of density. This is the most important point of this topic. Thank you. If you like my work, so please like, subscribe and share my channel.